Hello and welcome everybody to my channel Umini Bio Entrance, and today we will be discussing CSIR Net JRF Life Science question paper conducted in the year March 2025. This was actually to be conducted in December 2024, but they conducted it in March 2025. So this is the shift 1 or the morning shift question paper, and I am discussing the part B questions. I have not answered all the questions in part B, but questions from cell biology, genetics, molecular biology, applied biology and methodology, plant physiology, then cell biology, biochemistry, all these I have done. So first question, a novel organism synthesizes proteins using ribosomes from C-terminal end to the N-terminal end rather than the usual direction, that is N-terminus to C-terminus. Which main chain atom will be the nucleophile for the reaction to form peptide bonds? Now, you know, in the normal case, that is when it is synthesizing from the N-terminus to C-terminus, the nucleophile is your amino group of one amino acid. Why? Because it is having a lone pair of electrons on the nitrogen atom. And a nucleophile is generally, they have the ability to donate an electron pair. They are electron rich species, not electron poor. So the nitrogen atom, it has lone pair of electrons, and it is a good nucleophile. So this is the nucleophile of this particular amino acid. Now there is another species, that is the electrophile, which will accept these electrons and result in bond formation. Now the electrophile in the peptide bond formation is your carbonyl carbon, that is this carbon. I have written it in a small c over here, you can see it. So it is the carbonyl carbon of the other amino acid. So this is the second amino acid, and this is the electrophile. It is deficient in electrons and it will be attacked by the nucleophilic nitrogen. So that is what is happening normally. And there is liberation of water and the formation of the peptide bond. Now, the synthesis is reversed in this particular organism. How they have carried out the synthesis from C-terminus to N-terminus. Now, whatever be the direction of the synthesis of the amino acid, for the formation of peptide bond, somebody has to donate electrons and somebody has to accept it. And that donation of electrons is usually done by the nitrogen, because it is having lone pair of electrons. Whereas the carbonyl carbon of the other amino acid doesn't have electrons. It is deficient. So whichever may be the direction, nitrogen is always going to be your nucleophile. It cannot be the carbonyl carbon. Okay. So the answer here is nitrogen of the amine group. Answer is option 1. Then next one. The citric acid cycle operates only in the presence of molecular oxygen. Why? Because now you know, in the electron transport chain, electrons from NADH and FADH2, they are used to pump protons across the mitochondrial membrane, and this will result in the formation of the proton gradient, and this proton gradient, it will drive the synthesis of ATP, and oxygen will be the final electron acceptor. Now they have asked, if oxygen is not available for the electron transport chain to continue, then what will happen? Then these electron carriers, that is NADH and FADH2, they cannot be oxidized back to their original form, that is to NAD plus and FAD plus. It cannot be oxidized back, and without that NAD plus and FAD plus, citric acid cycle will not take place, because citric acid cycle needs NAD plus and FAD plus. It will not happen there. So why do you require molecular oxygen for the TCA cycle to operate? Is because oxygen accepts electron from the electron transport chain, allowing reoxidation of NADH to NAD plus. So 2 is the answer. Then the others, if you see, oxygen activating enzymatic dehydrogenation, no, that doesn't happen in a citric acid cycle. Then oxygen remove toxic products of TCA cycle, that is also not involved action of oxygen. Oxygen activates ATP synthase, never. It is the proton gradient that is activating the ATP synthase. So these 3 options are wrong, and the second is the right answer. Then which of the following statement is true regarding beta oxidation of fatty acids? So beta oxidation, it occurs, you know, in the mitochondrial matrix. So the first statement, it occurs in the intermembrane space region of mitochondria, is not correct. Then all reactions are same for saturated and unsaturated fatty acid. No, unsaturated fatty acid, they have double bonds. So they require additional step to remove these double bonds. So the reactions are not the same in saturated and unsaturated fatty acid. Then fatty acids are oxidized at C3 position to remove the 2 carbon atom. Yes. So beta oxidation, the carbon atom at the beta position is involved in the oxidation. So at the beta position means the carbon next to the alpha carbon atom. Now alpha carbon atom is the one which is close to the carboxyl end. So this is the carboxyl end. So the next one to alpha is your beta carbon atom. Now if you start numbering the carbon atoms, you start from here. So this is the first carbon, this is the second, and this is the third. So your beta carbon atom is at position 3. So option 3 is correct. So fatty acids are oxidized at C3 position to remove 2 carbon units. So answer is option 3. They have asked which is true. So lipoprotein lipase catalyzes the first step of beta oxidation. No, lipoprotein lipase is involved in the breakdown of triglycerides, releasing fatty acids and glycerols, and the first step of beta oxidation, it is catalyzed by acyl-CoA dehydrogenase. Then next one, which one of the following interactions will predominantly contribute to the stability of nucleosome? So nucleosome, it is a segment of DNA that is wrapped around the histone octamer. Okay, so these are the histone protein. This is the DNA that is wrapped around the histone protein. This is H1 protein. Now DNA, you know, it has negatively charged phosphate backbone, and histones, they are rich in arginine and lysine residues, which are positively charged amino acids. So histone is more of the positive charge and DNA is of the negative charge, and they both attract together, resulting in the wrapping of the DNA around the histone octamer. So there is positive charge and negative charge being involved. So it is electrostatic interaction that is taking place involving DNA phosphate backbone and lysine residues of histones. So 4 is the answer. It is not hydrogen van der Waals interactions. Then next one. Which of the following is likely to enter a pure phospholipid bilayer? So they have given the option, that is carbon dioxide and diethyl urea. So carbon dioxide, they are small hydrophobic molecules. So the membrane is easily permeable to them. Diethyl urea, it is also small hydrophobic molecule. So membrane is easily permeable. If you take water and glucose, water can move, it is mostly permeable, but glucose, it is a large uncharged and polar molecule. So it requires transport proteins. So second is not the answer. Lysine and ethanol, lysine will come under charged ions. It is charged group, so it will have difficulty in passing through. It requires transport proteins, whereas ethanol, it is small in size and it is hydrophobic, so it can easily pass. Then urea and chloride ions. Urea also it can pass, but chloride ions cannot pass. It is impermeable. It requires transport proteins. So out of these, the one which is more likely to enter the phospholipid bilayer is your carbon dioxide and diethyl urea. Answer is option 1. Then majority of modification of histones happen at there. So it is at the N-terminal tail of the histone that majority of modification like acetylation, methylation, phosphorylation, ubiquitination, all these happen. And N-terminal region, it is more accessible to the modifying enzymes because it is outside of the nucleosome core, it is located. Then which one of the following is not a major phospholipid? So major phospholipid in the mammalian plasma membrane. So the major phospholipid in mammalian, phospholipid I am telling, phospholipid in a mammalian plasma membrane is phosphatidylcholine, and phosphatidylinositol is the least one. It is a minor phospholipid in mammalian plasma membrane. So here you may think phosphatidyllinositol is the right answer, but cholesterol is more correct. Why? Because cholesterol is not a phospholipid. So it cannot be a major phospholipid at all. Okay. Even though it is found in significant amount in the plasma membrane. It is not a phospholipid at all. Then it is not the major phospholipid. So cholesterol is the answer. Then dynein is a microtubule dependent motor protein essential for cellular processes. Which of the following statement related to dynein function is incorrect? They have asked for the incorrect statement. It helps in cilia flagella beating. Of course, dynein does that. Then it transport organelles in anterograde direction. No, dynein is involved in retrograde transport of organelles. What is this retrograde means? So they move these organelles or proteins or anything from the plus end of the microtubule towards the minus end. Now plus end of the microtubule, it is found towards the cell periphery, and the minus end is usually towards the nucleus. So from the plus end to the minus end, that is retrograde transport, dynein is involved. Whereas in anterograde direction, it is the kinesin proteins that are involved, and they move these organelles from the minus end towards the plus end. That is from the nucleus to the cell periphery. Then next one. 2 types of mutant E. coli were identified in the hypermethylation mutant. Okay, hypermethylation means as soon as the newly synthesized DNA is produced, it will start methylating it very fast. Methylation. Okay. DNA is methylated at the GATC sequences as soon as daughter DNA is synthesized. That is hypermethylation. And the second type, GATC sequence, are never methylated. That is your type B. Now, which mutant will have greater effect on the MMR system, leading to accumulation of spontaneous mutation? Now, what is this MMR system? MMR is nothing but your methylation and mismatch repair system. So, whenever a newly synthesized daughter strand is produced, there may be mismatches. Now this mismatches, they are repaired by the MMR system so that mutations are reduced in the E. coli. That is what is MMR doing. So MMR has got a technique to identify between the parental and the daughter strands. So in bacteria, the parental strand is usually methylated, as you can see in this picture. This is the lower strand, it is methylated, CH3, you can see it is methylated. Whereas the upper strand, that is your daughter strand, which is non-methylated, or it will be hemi-methylated. Okay. Now these proteins present in the MMR machinery, they will recognize this hemimethylation and they will understand that it is the daughter strand that requires the repair because of the presence of mismatch, and they will start acting on the daughter strand. See, the daughter strand is cleaved over here. This MUTH is causing this nick. Then other mismatch repair proteins are involved. Then the DNA polymerase acts and synthesizes the new strand, and the newly synthesized strand is methylated after the action of joining by the DNA ligase. So that is what is happening in MMR. So here they have asked which mutant will have greater effect on the MMR leading to accumulation of spontaneous mutation. So if you take the case of type A, it is hypermethylation mutant. So as soon as the daughter strand is synthesized, it is methylated. So will the MMR system recognize this? No, they will have difficulty in recognizing between the parent and the daughter strand because both are methylated. How will they recognize and repair the mutation? It is not possible. So what will happen? In a type A, mutations will keep accumulating more. Whereas in type B, it is never methylated. So slow and steady. So type B, daughter strand is not methylated. Parent strand is methylated. MMR system can recognize if there is a mutation in the daughter strand, repair it, then allow methylation, and occurrence of spontaneous mutation is also less. So type B, the accumulation of spontaneous mutation will be less. Okay. Than in type A, which is hypermethylation mutant. So which mutant will have greater effect on the MMR? So it is type A will have greater effect than type B. So the answer is option 1. Then next one, E. coli genes in replication and functional eukaryotic orthologs. So orthologs, these are genes in one species. They have a homologous of these particular genes in another species. So here if they have given the prokaryotic genes involved in DNA replication and eukaryotic genes involved in DNA replication, so they both are orthologs. You have to find which is the ortholog of each gene which is involved in E. coli replication. So if you take the case of DNA B, you know it is a helicase involved in DNA replication in E. coli, and which is the one helicase that is involved in eukaryote, it is MCM complex. So A4, that means 1 gone and 4 also gone. 2 and 3 we have to see. A4. Okay. Now B will tell you the right answer out of 2 and 3. So next one is DNAC. You know it is the helicase loader protein in E. coli. Now in eukaryote, which is the helicase loader, that is the CDC6. So B2, that makes option 3 as the answer. Okay, option 3 will be your answer. Option 1, it is B1. So that will not be the answer. So CD6. Okay, CD6, it is a part of the pre-replicative complex, and it is involved for loading MCM complex onto the DNA in eukaryotes. Then beta clamp, it is for processivity of the DNA polymerase. And what is this processivity mean? It means that it will keep the DNA polymerase tightly attached to the DNA so that the DNA polymerase doesn't fall off from the DNA during replication. So that is processivity, and in eukaryote it is the pCNA that is involved in this processivity, so C3. Then DNAG, it is a primase, it is involved in the synthesis of RNA primers, which can help in the initiation of DNA synthesis by the DNA polymerase. So in eukaryote it is Pol alpha primase. Okay, so the answer is option 3. Then which of the following statement about RNA polymerase in eukaryotes is incorrect? You have to find the incorrect statement. So RNA polymerase 1 synthesizes 18S, 5.8S, 28S rRNA, that is 100% correct. Polymerase 2 messenger RNA, again correct. Polymerase 2 requires sigma factor to initiate transcription. No. Sigma factor is the one which is found in prokaryotic RNA polymerase. It is involved in promoter recognition and initiation of transcription in prokaryotes, not in eukaryotes. The question here is for eukaryotes, not prokaryotes. So that is the incorrect statement, and that is your answer. Whereas eukaryotic RNA polymerase, they require the general transcription factors like TF2D, TF2A, TF2B, etc. for initiation of transcription. So 3 is the answer. Then RNA polymerase 3 synthesizes 5S rRNA and tRNA is again correct.